Hello everyone. Let me introduce to the course Principles of Microbiology and Biochemistry. We have 4 course objectives, out of which the first one is that students should be able to characterize and cultivate the microorganisms. Students should be able to know the basics of sterilization techniques which are used for the pharmaceutical products, equipments and cultural media. And students should perform the microbial assays with respect to antibiotics and vitamins. They should also know the biochemical processes that happens in the human body, that is carbohydrate metabolism, carbohydrate synthesis, protein metabolism, and other like enzyme synthesis and its metabolism, lipid synthesis, its metabolism, and nucleic acid synthesis. After learning the course, students should be able to describe characterization and cultivation of microorganisms, and they should be able to describe sterilization processes, specifically what kind of sterilization is used for pharmaceutical products, what kind of sterilization is used for equipment and culture media. And they should be able to illustrate the sterility testing and microbial assay specifically with antibiotics and vitamins. And they should be able to organize biochemical processes like carbohydrate and protein synthesis and metabolism, as I mentioned, and enzymes, lipids, and nucleic acid synthesis and metabolism. So coming to the course structure of PMB, that is Principles of Microbiology and Biochemistry. This course has 5 units, and the first unit explains you about what is microorganism and how to cultivate the microorganism, and what are the different processes used for the preparation of media, and how to prepare a general media or specific media. All this which is related to the microorganism growth and factors influencing its growth, all this we will study in the first chapter. In the unit 2, we study about control of microorganisms by using various sterilization processes. We have physical sterilization process and chemical sterilization process. And in physical sterilization also we have various methods. In chemical sterilization we have various chemicals for the sterilization. Along with that we have one more sterilization process, that is radiation sterilization. All these things we will study in the second unit. In the unit 3, we will learn about the microbiological assays, which is specifically used for the vitamins and antibiotics. Along with that, we also learn the biological membrane of the human body. What is the composition of biological membrane? As you know, we have phospholipids in our membrane, cholesterol. What is the composition of each? That we learn in the unit 3. Coming to unit 4, in this chapter we study about metabolism of carbohydrates, proteins and nucleic acids. Coming to unit 5, we study the lipid metabolism and enzyme mechanisms. In unit 4, specifically we study about glycolysis, glycogenolysis, how the glucose is synthesized from the basic process, how the glucose is metabolized into simple components, and how the proteins are synthesized by the combination of amino acids, and how these proteins are converted into amino acids and urea and excreted through the human body. And coming to the nucleic acids, as you know, we have 2 basic nucleic acids, that is DNA and RNA. And we study specifically about central dogma, that is transcription, translation and post-translation changes that happens. And coming to the lipid metabolism, we study about beta reduction of lipids, alpha, beta and gamma reduction of lipids, metabolism of lipids. Along with that, we also study the synthesis of cholesterol. In enzyme mechanisms, we will learn about the enzyme activity, what factors will influence this enzyme activity, and also we learn about reversible and irreversible enzyme replacement at various sites. So the major expectations from the students during the learning process of this course is to actively participate in the lectures and discussions, and timely submission of assignments and projects, regular attendance and engagement their involvement in the classes and lab activities as well, consistent preparation for the assessments. Coming to the textbooks, for microbiology part, you can refer Pelzer's microbiology book, and for biochemistry part, you can refer a book written by U. Satyanarayana, which are available in our library. Thank you for listening.
Vishnu Audio Visual Center, like and share and subscribe and click bell icon.